Have you ever stopped to think about how scientists actually get a handle on what's happening inside a massive, sprawling ecosystem? I mean, it's a huge, huge challenge. It's not like you can go out and count every single blade of grass or every leaf on every tree. So, how in the world do they get a clear picture?
Right? I mean, how do you measure a meadow, or a whole forest, or a sprawling coral reef? This is like the fundamental question ecologists wrestle with every single day as they're trying to monitor the health of our planet.
Well, to get around this, they came up with this really key metric called species cover, and it's a simple idea, but it's super powerful. Basically, imagine you're a bird flying way up high looking straight down. What percentage of the ground is covered by, say, dandelions? That single number, that percentage, it tells you a ton about the ecosystem's health and how diverse it is.
Okay, but that still kind of leaves us with the big question, right? How do you measure that percentage without measuring, well, everything? Well, for decades, ecologists have had this really clever tool up their sleeves that lets them do something amazing. Count without actually counting.
It's called the point-intercept method. And the idea is it's brilliant in its simplicity. See, instead of trying to measure the whole area, you just sample a small number of individual points. It's kind of like a political poll, You don't have to ask every single person in the country who they're voting for. You just ask a well-chosen sample, and you can get a surprisingly accurate picture of the whole country.
And just to be clear, this isn't some newfangled idea. This method has been the gold standard for a long, long time. And the reason why is that it's just so objective. It takes all the guesswork, all the human bias right out of the equation.
So, how does this actually work when you're out in the field? Okay, picture this. A scientist walks out with this square frame thing, it's called a quadrat, and they just place it down randomly. Then, at every little intersection point on that frame's grid, they lower a tiny pin straight down. If the tip of that pin touches a plant, boom, they write down what it is. So, if, say, 30 of their 100 pin drops hit a certain type of grass, they can be pretty confident that grass has about 30% cover in that area. Pretty simple, right?
And this is what that data actually looks like on a field sheet. Every single one of those little tick marks is a hit. One moment in time where a pin touched a plant. I know it looks a little chaotic, right? But every single one of those marks is a crucial data point. And when you combine it with hundreds or even thousands of others, you start to build this incredibly precise statistical picture of the entire landscape.
All right, but here's where the story gets really interesting. For all of its power, this trusted method had a huge hidden flaw. It was kind of like having a great recipe that was missing all the measurements, and it was threatening to throw off decades of important research.
See, the problem wasn't the tool itself. It was the instructions, or rather, the lack of them. Without any standard way to do things, scientists all over the world were using the same basic method, but in totally different ways.
Think about it like this. Maybe one group of scientists uses a frame with 100 points all packed tightly together. But another group, studying the exact same field, might use a much bigger frame with only 36 points spaced way far apart. They could get totally different results, and nobody would know which one was right. It was a complete apples to oranges comparison nightmare. And this really gets to the heart of the problem. As the research paper points out, scientists were often just doing things based on past practice. Basically, they were following tradition, doing it the way their mentors did it, instead of using a protocol that was backed up by hard math. So that leaves this massive question hanging in the air for the entire scientific community. If everybody's using the tool a little bit differently, how do you find the one best way to do it? How do you finally write the official rulebook? So what they did was, honestly, it was brilliant. They decided to do something you could never do in the real world. They took the entire messy, complicated, unpredictable field, and they built it inside a computer. Now, here's the absolute genius of this. They built a perfect digital ecosystem, a world where they knew with 100% certainty the exact species cover, right down to the last digital pixel. In this perfect little world, the true answer was a known thing, which is something you can never have in a real meadow. So once they had their perfect digital meadow, they basically sent in an army of virtual scientists to sample it over and over and over again. They ran thousands of simulations, testing every combination of point spacing and frame sizes you can imagine, all to see which method consistently got them closest to that known, true answer. And when I say they did this a lot, I mean a lot. In total, they ran simulations for 72 million quadrats.
That is a staggering amount of computing power, all focused on finding one simple rule that had been hiding this whole time.
And after all of that, after wading through the data from 72 million virtual tests, it all came down to one single elegant guideline, a rule that would change everything. The magic number? 80%. That was it. That was the aha moment, the key that unlocked the whole thing. So with this one number, researchers could finally write the instruction manual that had been missing for decades. A new set of rules to make sure everyone, everywhere, was collecting data that was consistent, reliable, and could actually lead to better decisions.
So what is the 80% rule? It is beautifully simple. It just says that to get accurate results, the space between your sampling points has to be at least 80% of the diameter of the biggest plant you're measuring. That's it. So if the widest shrub you're looking at is a meter across, your points have to be at least 80 centimeters apart. That's the rule.
And this one simple rule basically created a whole new standardized playbook for scientists. Now they can figure out exactly how many points they need before they even get out there. They can make sure their spacing is right. And, crucially, they can report the exact margin of error for their findings, which makes the science so much more transparent and trustworthy for everybody.
Okay, so why should we care about some statistical rule for scientists poking pins in the ground? Because this matters a lot. Having better, more reliable data is the foundation for, well, everything. It means we get smarter conservation strategies. It means we can track the effects of climate change with way more accuracy. And it means we can make better decisions about how we manage our planet.
I mean, it's just an incredible story of how a massive computer simulation solved this decades-old problem. And it really makes you wonder, doesn't it? What other classic scientific tools, in any field you can think of, are just one clever virtual test away from a massive breakthrough?